Good afternoon, students. So, today we will continue with this chapter, that is human reproduction, and the next topic is the post fertilization changes. Now, what happens after the fertilization? You know that the zygote is formed as a result of fertilization. Then comes the segmentation of this zygote, which is known as cleavage. So, what is cleavage? This term refers to a series of rapid mitotic divisions of the zygote following fertilization, forming a many-celled blastula. Now, here you can see the differences between a cleavage and regular mitosis. Now, what is the difference? Now, cleavage occurs in the zygote or parthenogenetic egg, whereas mitosis occurs in most of the body cells. In cleavage, the interphase is short and blastomeres do not grow during it, whereas the interphase is long and daughter cells grow to normal size during mitosis. The size of the blastomeres decreases as their number increases in cleavage, whereas the size of the daughter cells they remain nearly constant due to growth in typical mitosis. The total mass of blastomeres remains the same as that of the zygote, whereas in mitosis the total mass of the daughter cells becomes more than that of the parent cell. DNA synthesis occurs much faster to form chromosomes for new blastomeres, whereas the DNA synthesis occurs at the normal rate in mitosis. The nuclear cytoplasmic ratio increases as cleavage progresses, whereas the nucleus and the cytoplasmic ratio does not change in mitosis. So, is that clear? This is the difference between the division which is known as a cleavage or mitosis, how we can differentiate the two. Next, we come to the cleavage in the human zygote. Here you can see on your screens is displayed the diagrams, the different stages of the cleavage up to the formation of the early morula. So, cleavage in the human zygote occurs during its passage through the fallopian tube towards the uterus. It is holoblastic, that is, it divides the zygote and blastomeres completely into daughter cells. However, the blastomeres differ in size from the start. Their number does not increase by a regular doubling sequence. The first cleavage takes place about 30 hours after fertilization. It is meridional, coinciding with the animal vegetal pole axis. It produces two blastomeres, one slightly larger than the other. The two blastomeres remain adhered to each other, that is, they stick to each other. The second cleavage occurs within 60 hours after fertilization. It is at right angles to the plane of the first and divides each blastomere into two by forming a mitotic spindle in each. The larger blastomere divides a little sooner than the smaller one, so that there is an intermediate 3-celled stage before the 4-celled stage of the embryo. Third cleavage takes place about 72 hours after fertilization. Subsequent cleavage divisions follow one after another in an orderly manner, but in a less precise orientation. Cleavage produces a solid ball of small blastomeres. So, now the embryo looks like a mulberry, so it is known as morula stage. Morula means mulberry in Latin. It refers to the lobed surface of the embryo at this stage. Cleavage is radial and intermediate in human zygote. In radial cleavage, At 8 cell stage, the upper 4 cells are directly above the lower 4 cells. In indeterminate cleavage, each blastomere formed by the early cleavage divisions retains the capacity to develop into a complete embryo. It is the indeterminate cleavage of the human zygote that makes identical twins possible. Is that clear? This 8-shaped structure. So this is very important event which occurs during this early mammalian cleavage. This occurs at the 8 cell stage. Here the blastomers, which are loosely held, they tightly adhere to one another, and the proteins which adhere them to one another or attach them to one another, they are known as the cohesions. Now, what is the significance of cleavage? The cleavage restores the cell size and nucleocytoplasmic ratio characteristic of the species. It does not result in growth, though it increases the number of cells. During cleavage, cellular activity is still mainly controlled by the organelles and molecules received from the secondary oocyte cytoplasm. But some of the developing organisms' genes become active. Cleavage, besides producing a large number of cells by rapid divisions, also segregates different substances present in the cytoplasm into different cells. These substances determine how the various cells are going to develop later on. Cleavage produces a large number of cells, so the building of the offspring's body may begin. Right? So then is the next stage, that is the morula, which you can see here in the last diagram. That is the early morula. You see all the stages, the early zygote, then 2 cells, then the 3 cell, 4 cell stage, and 8 cell stage also, and after 8 cell stage is the early morula. Now about the morula and the blastula, that is the morula and the blastocyst. So what is morula now? The morula, as I told you earlier, is a mulberry like structure. The morula consists of 16 to 32 cells, but it is not larger than the size of the zygote. Its cells are of 2 types. There are an outer layer of smaller, clearer cells around an inner mass of larger cells, as you can see in the diagram here. The morula reaches the uterus about 4 to 6 days after fertilization. It is still surrounded by zona pellucida that prevents its sticking to the uterine wall, and you can see this clearly in the diagram displayed on your screens. Now what about blastocyst? The outer cells of the morula now absorb the nutritive fluid secreted by the uterine mucous membrane and is known as the trophoblast or the trophoectoderm. The cells of the trophoblast become flattened. The fluid absorbed by the trophoblast collects in a cavity, the blastocele or blastocyst cavity, that separates the trophoblast from the inner mass cells except at one side. As the quantity of fluid increases, the morula enlarges rapidly and assumes the shape of a cyst. Then it is known as blastocyst. Inner mass cells now look like a small knob at one pole. The knob gives rise to the embryo and is known as the embryonal knob. You can see it very clearly in the second diagram displayed on your screens, the B diagram. The side of the blastocyst to which embryonal knob is attached is known as the embryonic or animal pole, and the opposite side is the embryonic pole. So one is the embryonic pole, other is the abembryonic pole. The trophoblast does not take part in the formation of the embryo proper. It remains external to the embryo and gives rise to the extraembryonic membranes, namely chorion and amnion. So for the production and nourishment of the embryo, the trophoblast cells in contact with the embryonal knob are known as cells of Robber. As the blastocyst is formed, zona pellucida becomes thinner and finally disappears. So in the second diagram you do not see the presence of zona pellucida. You can see the embryonal knob, you can see the trophocele, and you can see the embryonic pole and the abembryonic pole. Both these poles you can see, and you can see the blastocele, that is the fluid filled cavity. So next, now what we are going to talk about is the implantation. Up till here I hope everything is clear to you. So now we talk about implantation. So what is implantation? It is the attachment of the blastocyst to the uterine wall. It takes place about 7 days after fertilization. So it takes 7 days for implantation to take place. Here you can see the stages of human development from fertilization up to implantation in the diagram. So the blastocyst, which has over 100 cells, from the portion where embryonal knob is located, lies against the endometrium. This contact stimulates rapid divisions of the trophoblastic cells, which destroy the endometrial cells by releasing lytic enzymes and engulf the eroded cells. The blastocyst sinks into a pit formed in the endometrium and then gets completely buried in the endometrium that grows around it. Okay? Do you get it? So here in this diagram you can see the pit in the endometrium. Up to 7th day you see from the first day after the entry of this ovum into the fallopian tube, you see the fertilization and then the cleavage first day, then the 3rd day is shown, and then the 7th day is shown. I hope it is clear to you. So we can move to the next diagram for the formation, this implantation, and then the formation of the germ layers. So thus implantation involves the activities of both the blastocyst and the uterus. The embedded blastocyst forms villi all around it to obtain nourishment. So this establishes a parasitic relationship between the embryo and the uterus, that is the mother. Then the embryo projects into the uterine cavity and loses villi from the free surface. Now what is the function of the zona pellucida? It is to prevent the implantation of the blastocyst at an abnormal site. It does not expose the sticky and phagocytic trophoblast cells till the blastocyst reaches the proper implantation site. Then the trophoblast of the invading blastocyst develops the rudiment of chorion, an extraembryonic membrane which later forms the placenta. The chorionic cells secrete a hormone which is known as HCG, that is human chorionic gonadotrophin. This resembles and takes over the job of the pituitary luteinizing hormone, that is LH, during the pregnancy. So the HCG maintains the corpus luteum and stimulates it to secrete progesterone. which is the essential hormone for the maintenance of the pregnancy. The progesterone maintains the endometrial lining and causes it to grow throughout the pregnancy. This prevents the menstruation during pregnancy. Thus, the blastocyst ensures its own survival, because if menstruation occurs, the developing offspring can be eliminated from the mother's body along with the endometrium lining of the uterus. So, by the 16th week of pregnancy, the placenta itself produces enough progesterone and the corpus luteum, and which is now unnecessary, regresses. Progesterone causes increased secretion of mucus in the cervix that forms a protective plug during the pregnancy. Now, about the different layers, that is the transformation of the blastula into gastrula with the primary germ layers by the rearrangement of the cells, and this process is known as gastrulation. So here we come to the next step, that is the gastrulation, which is also known as the formation of the germ layers, right? So the germ layers are formed by characteristic movements of some of the cells at or near the surface of the embryonal knob to their new, more inner locations. These cellular movements are known as morphogenetic movements. Cell movement is accompanied by changes in the cell shape and changes in cellular adhesion to the cells. There are three germ layers: ectoderm, mesoderm, and endoderm. They serve as an initial rudiments of all the future tissues and organs of the offspring's body. The germ layers give rise to specific tissues, organs, and organ systems of the young one to be developed. So this is about the germ layers, and you can see in the diagrams displayed, you can see clearly how the amnion, the chorion, the ectoderm, mesoderm, and the endoderm are shown, where the this yolk sac is present, where the extraembryonic mesoderm is present. So you can see all these in position and of the different layers after the implantation, and how it is embedded in the uterine wall. Next we come down to organogenesis. Organogenesis is not in detail in your syllabus. So we talk about organogenesis as the establishment of germ layers, which initiates the final phase of embryonic development. It involves differentiation and specialization of groups of cells in the individual germ layers. So that is how different organs are formed. Organogenesis means the formation of different organs from ectoderm, mesoderm, and endoderm. Is that clear? You can see here. You can see the fetal membranes and the placenta. So next, now we'll talk about placenta. So placenta serves to bring the fetal and the maternal blood close enough to permit exchange of materials between the two. A fully formed human placenta is a reddish brown disc about the size of a dinner plate, and it is less than 1 kg in weight. Its fetal surface is smooth and has an umbilical cord attached near its center. You can see it in the diagram. It consists of chorion only, hence it is called chorionic placenta. Allantois remains small and the allantoic blood vessels, however, extend into vascularize the placenta. Right? So, in the placenta, a large number of villi from the vascular chorion penetrate the corresponding pits, the crypts, which are formed in the uterine wall. The latter becomes very thick and highly vascular to receive the villi. The intimate connection established between the fetal membrane and the uterine wall is known as hemochorial placenta. The placenta has two parts: the part controlled by the fetus, that is the chorionic villi, known as the fetal placenta, and the part shared by the mother, that is part of the uterine wall, is known as maternal placenta. The chorionic villi come to lie in uterine lacunae that contains blood. The cells forming the wall of chorionic villi bear microvilli which increase their surface area for quick and proper exchange of materials by diffusion, active transportation and pinocytosis. Now, in the placenta, the fetal blood comes very close to the maternal blood, and this permits exchange of materials between the two. Food, that is glucose, amino acids, simple proteins, lipids, water, mineral salts, vitamins, hormones, antibodies and oxygen pass from the maternal blood into the fetal blood, and fetal metabolic waste such as carbon dioxide and urea, also water, hormones, they pass into the maternal blood. The placenta serves as a nutritive, respiratory and excretory organ of the fetus. The maternal and the fetal blood are not in direct contact in the placenta. Why? Because the two may be incompatible. The pressure of maternal blood is far too high for the fetal blood vessels, and there should be a check on the passage of harmful materials from like these germs or blood proteins from the mother into the fetal blood. So what are the main functions of placenta? Main functions of placenta are to supply the from the mother's blood into the fetal blood. So it is sort of a nutritive organ. It is also a digestive organ because the trophoblast of the placenta digests proteins before passing them into the fetal blood. It is a respiratory organ because oxygen diffuses from the maternal blood into the fetal blood through the placenta. Carbon dioxide diffuses from the fetal blood into the maternal blood also through the placenta and is eliminated from the mother's lungs. Then as an excretory organ, the nitrogenous waste such as the urea, they pass from the fetal blood into the maternal blood via the placenta and is eliminated from the mother's body from the kidneys. Then it also acts as an endocrine organ. Placenta secretes the human chorionic gonadotropin, estrogen, progesterone and human placental lactogen. Right? So all these hormones, they are secreted by the placenta. Then it is also as a storage organ. It stores glycogen for the fetus before liver is formed, and it also acts as a barrier for many substances which pass from the mother to the fetus. Such substances, they are known as teratogens. They may be some chemicals or viruses and the this dangerous synthetic teratogen drug is thalidomide, which causes multiple defects in the embryo. Even nicotine from cigarette smoke crosses the placenta and stunts the fetal growth, so expectant mothers should not take nicotine. Then addictive drugs like heroin and cocaine, they reach the fetus, they cause addiction to the newborn. So expectant mothers or the pregnant women should avoid all these things which are going to harm the fetus. They harm the or hamper the normal development of the fetus. The viruses and bacteria also cross the placenta, but this is offset by certain antibodies, globulins, certain antitoxins which pass from the mother to the fetus. Now the last part of this chapter is parturition and lactation. Parturition is the act of expelling the full term young one from the mother's uterus at the end of the gestation, right? And lactation is the production of milk in the female's breast following the birth of a young one in mammals. This diagram displayed on your screens shows the hormonal reactions during the labor, which hormones they are released during the parturition, that is the labor. So here you can see the estrogen from the ovaries, it induces oxytocin receptors to the uterus. Oxytocin from fetus and mother's posterior pituitary, it stimulates the uterus to contract, and it stimulates the placenta to make the prostaglandins. It stimulates more contractions of the uterus, and then they again stimulate, the positive feedback is for the placenta to make more these prostaglandins. Now this is the feedback control which is positive in nature, and it can also induce, it will go back to oxytocin, and this oxytocin will again it will stimulate the uterus to contract. So that is why this oxytocin is also known as the birth hormone. So conclude, I'll tell you that the average duration of human pregnancy is around 9 months, and this time period is known as the gestation period. There are vigorous contractions of the uterus at the end of this period, and it causes the expulsion or the delivery of the fetus. This is what I told you is known as parturition. Parturition is induced by complex neuroendocrine mechanism. The signals for parturition originate from the fully developed fetus and the placenta, which induce mild uterine contractions, which are known as fetal ejection reflex. And this of course triggers by the release of oxytocin from the maternal pituitary, as I just told you from this diagram which is displayed on your screens. The stimulatory reflex between the uterine contraction and oxytocin secretion continues, that is the positive feedback continues, which results in a stronger and stronger contraction. This leads to expulsion of the baby out of the uterus through the birth canal, right? And soon after the delivery of the infant, the placenta is also expelled out of the uterus. Now lactation. The mammary glands of the female undergo differentiation during pregnancy, and they start producing milk towards the end of pregnancy by the process which is known as lactation. This helps the mother in feeding the newborn child. The milk produced during the initial few days of lactation is known as colostrum, and this colostrum contains several antibodies absolutely essential to the developed resistance for the newborn baby. So, breastfeeding during the initial period of infant growth is recommended by the doctors for bringing up of a healthy baby because it develops the strong immunity in the newly born. So, with this, we end up with this chapter. Okay, students, I hope you understood it well. Any doubts, please text me and just look up into your assignment and turn in within the stipulated time. Thank you. Have a nice day.